Hi guys, welcome to my YouTube channel. In this video, we will learn about Pseudomonas aeruginosa. It is virulence factors, infections, laboratory diagnosis, and the best therapy options. Pseudomonas aeruginosa is a gram-negative rose typically arranged in pairs. It's notably characteristic by mucoid polysaccharide capsule. It has simple nutritional requirements and is capable of growing in a wide range of environments. This bacterium is an obligate aerobe and oxidizes glucose for metabolism. The oxidase test is positive in this species, which is a very important characteristic, something we will explore further when discussing how it helps differentiate Pseudomonas from other bacteria in the laboratory. Pseudomonas aeruginosa can grow on McConkey agar and form as colorless colonies. It can also produce colonies with a characteristic green fluorescent pigment on the culture media. Write for me in comments what is the reason behind the green fluorescent coloration produced by Pseudomonas aeruginosa on culture media. Pseudomonas aeruginosa is a very powerful bacterium. It causes serious and life-threatening infection. It is one of the predominant bacteria in both hospital and community-acquired infections. It has multiple factors which help it for inducting an infection. First of all, we talk about its adhesins. Aeruginosa have several adhesins, including flagella, alginate capsule, lipopolysaccharide, and pili. We know about LPS. Aeruginosa has a LPS like other gram-negative bacteria, but we have an important factor, alginate capsule. Alginate is a mucoid exopolysaccharide that forms a prominent capsule on the bacterial surface and protects the organism from phagocytosis and antibiotic killing. And about flagella and pili. Many students think are same, but they have several differences, especially in the function. Pili are crucial for initial stages of infection. They help bacteria adhere to host cells and tissues, which is the first step of colonization. Type 4 pili play a role in biofilm formation, which contributes to chronic infection. And also 6 pili allow for the exchange of genetic material, including genes for antibiotic resistant. On the other hand, flagella enable bacteria to move toward or away from certain stimuli, which helps them negative toward favorable environments such as nutrition or away from hostile environments such as toxins. Flagella also contribute to virulence by aiding the bacteria in escaping immune response and infecting tissues. Pseudomonas aeruginosa also has secreted toxin and enzymes. Several toxin and enzymes like exotoxin A, proteases, phospholipase C, elastase, exoenzyme S and T, pyocyanin, and pyoverdin. Don't worry, we will discuss all of these in this video. Pseudomonas aeruginosa has a type 3 secretion system. In summary, secretion systems injected particular materials from the bacterial cell to the host cells. In this bacteria, this system plays very important roles, including its role in antibiotic resistance and toxin secretion. Here, we can see a structure of this secretion system. In this part, in bacterial cytoblasts, we can see toxins have been produced and are waiting for secretion through the system. And we can see here the structure of secretion system. This system have many parts that have a role in this translocation, but we have here four main part of this system. PSCI have a role responsible for assembly of secretion needle. PSCF assessing to transport of substrates, and PCRV is a regulatory protein that plays a critical role in the assembly of the type 3 secretion system needle complex, which is a part of the larger translocon structure that injects effector protein into the host cell. And finally, we can see two component system of this secretion system, POP-B and POP-D. In this system, actually two proteins work together to form a translocon that allows effector proteins like exo-S, exo-U, and other exotoxin to enter to the host cells. Earlier, we talked about secretion system 3 that injected 4 toxin into the host cells. Now, we want to talk about effect of these toxin in human cells. First of all, we talk about exoenzyme S and exoenzyme T. These toxins are bifunctional enzyme with GTPase and ADP ribosylase transferase activity. We will discuss about ADP ribosyl transferase activity in more detail later. But in total, one of the key targets of this toxin is RHO family GTPase. By modifying this GTPase, exo-T and exo-S disrupts the regulation of the actin cytoskeleton and cell signaling, contributing to host cell damage and immunosuppression. Second toxin is exoenzyme U. This toxin is a phospholipid, which are the major component of cellular membranes. By breaking down these phospholipids, exo-U disrupts the integrity of the host cell membrane, leading to cell lysis and death. And the last toxin is exoenzyme Y. This toxin is an adenylate cyclase. Exo-Y functions as a calmodulin-dependent adenylate cyclase. It increases cyclic AMP levels in the host cells by catalyzing the conversion of ATP to cAMP. The increase in cAMP levels disrupts normal cellular signaling and can lead to alterations in host cell function. Elevated cAMP levels affect various cellular processes, including ion transport, immune response regulation, and cellular metabolism. In summary, when the type 3 secretion system introduces the proteins into their target eukaryote cells, epithelial cell damage occurs, facilitating bacterial spread, tissue invasion, and necrosis. This cytotoxicity is mediated by actin rearrangement. And finally, we want to talk about the most important and the most destructive toxin of Pseudomonas aeruginosa, exotoxin A. First of all, it is important to note that this toxin, unlike other exotoxins like exo-S or exo-T, not secreted by the type 3 secretion system. Instead, it's secreted through the type 2 secretion system. And its toxin is very likely to the diphtheria toxin produced by Corynebacterium diphtheria. Exotoxin A is an AB model toxin. It means it has two parts. Part B for introducing toxin to the host cell through receptor-mediated endocytosis, and Part A. Part A has enzymatic activity. This toxin is chromosomal. That means it codes in chromosome of the bacteria, not other genetic elements like plasmid. Receptor of toxin is alpha-2 macroglobulin. Is a large plasma protein found in blood. And this toxin have several effects in human cell, like inhibit phagocytosis, damage respiratory epithelial tissue, and inhibits release of inflammatory cytokines like tumor necrosis factor alpha and beta, interleukin 1 alpha and beta, and interferon from both macrophages and lymphocytes. And but how it causes this destructive effect in human cell? This toxin have a ADP regulating activity. It means toxin give a ADP ribose from nicotine dinucleotide and add that to elongation factor 2. Elongation factor 2 is a key protein required for ribosomal function during translation that cause shut up the peptide chain elongation and finally protein synthesis. That means this toxin interrupting the protein synthesis. This is a very important toxin. Several role in a human in a disease caused by Pseudomonas, like it contributes to dermonecrosis that occurs in burn wounds, coronal damage in ocular infection, and tissue damage in chronic pulmonary infection. Up to this point, we have discussed various important toxins of Pseudomonas aeruginosa, including its exoenzyme, exotoxin A, secretion system structure, and pathogens. Now, we would like to talk about other viral factors such as elastase. Pseudomonas aeruginosa has two elastase. Elastase A act as a serine protease and elastase B act as a zinc metalloprotease. These elastase act synergistically to degrade elastin, resulting in damage to elastin-containing tissues and producing the lung damage and hemorrhagic lesions known as ecthyma gangrenosome associated with disseminated Pseudomonas aeruginosa infections. Second factor is phospholipase C. This factor is a heat label hemolysin that breaks down lipids and lectin, facilitating tissue destruction. The exact role of this enzyme in respiratory and urinary tract infection is unclear. Also, an important association between hemolysin production and disease has been recognized. Many other bacteria has this enzyme, like as Clostridium perfringens, Listeria monocytogenes, or Mycobacterium tuberculosis. Next factor is a pyocyanin. Is a black pigment produced by Pseudomonas aeruginosa, catalyze the production of superoxide and hydrogen peroxide, toxic form of oxygen. This pigment also stimulates interleukin 8 release, leading to enhanced attraction of neutrophils. And last factor is pyroverdine, a yellow-green pigment. Is a pseudophore that bends iron for use in metabolism. This pigment also regulates secretion of other virus factors like exotoxin A. Let's talk about laboratory tests for detection Pseudomonas aeruginosa. Pseudomonas aeruginosa in the smear show as a gram-negative rows. For culture, we can culture Pseudomonas aeruginosa in various media like McConkey agar or Muller-Hinton agar or blood agar. And culturing on this media usually does not require any special incubation condition. An incubation temperature of 37 under aerobic condition for 12 to 18 hours is sufficient for growth of Pseudomonas. One important point about the growth of this bacterium in the McConkey agar is that Pseudomonas forms colorless colonies in this media. This is because Pseudomonas is unable to ferment lactose. In contrast, bacteria such as Klebsiella or Escherichia coli, which can ferment lactose, produce pink-colored colonies on McConkey agar. In biochemical tests, the oxidase test is a positive in Pseudomonas aeruginosa, which is a very important diagnostic factor. This is because other clinically significant gram-negative bacteria we often encounter in the lab, such as member of Enterobacteriaceae family, as Enterobacter baumannii and Strophomonas maltophilia, are oxidase negative. Therefore, this simple test can effectively differentiate Pseudomonas from these other important gram-negative organisms. About TSI test, in TSI, Pseudomonas produces a red color both on the slant and on the butt of the tube. This result indicates that the bacterium does not ferment any sugars. These characteristics make it easy to distinguish Pseudomonas from members of Enterobacteriaceae family using a simple test. Because Enterobacteriaceae typically ferment sugars at least on the slant portion of the tube. And about odor, colony of Pseudomonas aeruginosa often have a grape-like odor. So let's talk about clinical diseases caused by Pseudomonas aeruginosa. First important infection about Pseudomonas aeruginosa is pulmonary infection caused by Pseudomonas aeruginosa. Aeruginosa infection of the lower respiratory tract can range in severity from asymptomatic colonization or benign inflammation of the bronch to severe necrotizing bronchopneumonia. Colonization is seen in patients with cystic fibrosis or neutropenia. The mortality rate is high as a 70%. Second infection is UTI. Urinary tract infection is seen primarily in patients with long-term urinary catheters. And about air infection, external otitis is frequently caused by Pseudomonas aeruginosa, with swimming an important risk factor, known as a swimmer's air. The localized infection can be managed with typical antibiotics. And about primary skin and soft tissue infection. Aeruginosa can cause a variety of primary skin infection. The most recognized are infection of burn wounds, colonization of the burn wounds followed by localized vascular damage, tissue necrosis, and ultimately bacteremia is common in patients with severe burns. Folliculitis is another common infection caused by Pseudomonas resulting from immersion in contaminated water like hot tubs or swimming pools. Pseudomonas endocarditis is uncommon and is primarily seen in intravenous drug abusers. This patient acquire the infection from the use of drug paraphernalia contaminated with waterborne organisms. And bacteremia caused by Pseudomonas aeruginosa is clinically indistinguishable from that caused by other gram-negative bacteria. However, the mortality rate in affected patients is higher with Pseudomonas bacteremia because of number 1, pre-allocated of organisms for immunocompromised patients, number 2, difficult in treating antibiotic-resistant strains, and number 3, the inherent virulence of Pseudomonas. Thanks for watching this video until the end. I hope the content was helpful and informative for you. To see more educational videos like this, please subscribe to the channel. If you have any questions, suggestions, or feedback, feel free to share them in the comment section. Until next time, stay safe and keep learning.